Okay, so today's video covers mitosis and meiosis. Mitosis is generally what happens when your cells divide, and meiosis is only occurring when your haploid sex cells divide, the ones that you need for reproduction. So in the grand scheme of the cell cycle, mitosis is actually not where the cell actually spends most of its time. And if you've read ahead, there are certain stages of mitosis even that can subdivide. There's actually a G1 stage, it's a G0 stage, and in these phases, the cell's growing, it's getting nutrients, and there are certain signals that tell it to go on to, for example, S phase. This is where the chromosomes double in advance of mitosis, and G2 phase, kind of the final checks on the chromosome. So each of these checkpoints represent a spot where the cell can decide whether it wants to divide or not. If there's DNA damage or something, usually these checkpoints cause the cell to die. In the case of cancer, though, for example, the cell just keeps going through this cycle over and over and over. So the basic idea with mitosis is that, so for a single chromosome pair, so this is the same chromosome, you have one from mom and you have one from dad. So you have a pair of chromosomes. So preceding mitosis, the one from mom is doubled, the one from dad is doubled. So now you have 4 total of the same chromosome, even though you have 2 variations, remember, maternal and paternal. So then these are actually pulled 2 different ways by the dividing cell. And ultimately, you're gonna have 1 maternal, 1 paternal, 1 maternal, and again, 1 paternal. Dad, mom, dad. But now I see you have 2 cells. So that's your basic overview. Sorry, I didn't leave that up long enough. I might have to pause the video right there. So within mitosis itself, there are 1, 2, 3, 4, 5 distinct stages that when you know, you can diagnose what happened in a cell. So for example, I can tell when something goes wrong at metaphase if certain chromosomes aren't aligned properly and you accidentally gain one or the other. So let's just start with interphase. So this is before mitosis essentially. And in this state, the cell has its membrane, it has its nucleus, a chromosome pair is just sitting there, it's not doing anything. We'll do 2 at a time. Prophase. In this, the major step is that the chromosomes start to condense, and they get really dense in advance of being divided. And not only that, the nuclear membrane starts to dissolve and break, and there are certain factors that do this. So now there's this nuclear membrane is going to release so that you can get at the chromosomes. Not only that, the final thing is that protein complexes form what we call the spindle at both sides of the cell. And this is the first chance that they have. The spindle is important because it's what's going to pull the chromosomes apart. And what you have are actually microtubules. So these are just built of little units like this, just single units, but they can stretch really long. And what they do is they're going to be the ones pulling and pushing the chromosome. So at prometaphase, this is before everything's aligned. The nuclear membrane has now fully gone away. And each of the chromosomes, remember, has their own pair. So this is 2 of the same chromosome, 2 chromatids. So those are now condensed and free. What happens is, remember we have our spindles, and they're little microtubules. Here, I'll make this a little clearer. Let's see how I drew this, yeah. So these microtubules now, what they're doing is they're going back and forth, and they're eventually attaching to the centromere, which is connecting those 2 chromatids. To do this though, they actually add, and if they miss, they delete their microtubules and the subunits disassemble and then they eventually hit them. And so they connect to the kinetochores, and that's what's kind of in the centromere here, and that's what's like kind of the little motor that's going to tear the chromosomes apart. So not only that, but there are microtubules that attach to the outside of the cell here, called astral. The ones that are attached to the chromosomes are called kinetochore. And there are also microtubules that just attach to each other and form an area of push. So if we just do metaphase, this is right before the cell divides. So here's our spindle. Now let's see, I'm gonna draw this. Yeah, sorry. So here they've attached and they're getting closer. And here, everything is attached. Everything is ready to divide. Now the cell, remember there's pushes here, these polar microtubules. So the cell's ready and it's set, the chromosomes are set right on the plate, and this is where it's gonna split off like that. These chromatids are gonna get pulled. And remember, these are identical, so you're not losing any genetic information. Oops. Okay, so anaphase is where the pull comes. So at this stage, let's say we have our cell's getting a little bigger. So we have our spindles, and what they've done is that they've ripped the chromatids from each other. So remember, these were together, but they've been pulled away, and it does it really quickly. It does it with the help of the microtubules here that are pulling, the astral microtubules here that are pulling this way, and then the polar microtubules that are touching each other from each thing, and these are pushing. These are gaining and pushing. Oh yeah, I'll do two. So then in telophase or telophase, here I'm going to do it from this angle. So cytokinesis is kind of happening here. The chromosomes, they go from this coiled state back to their uncoiled state. And the nuclear membrane starts to reform. So these are small characteristics that kind of can define each cell, and just as well, or each cell phase, I should say. And then the spindles, remember, these are on this side, they're starting to disassemble too. And the final stage is that they actually split apart, nuclear membrane reforms, and now you have two identical cells to the one that we started with. There's some really good videos out there, like with animations and stuff, so you can see like the real deal with them. So meiosis is a little different because